The sense organs, the first thing we need to talk about are receptors. A receptor is a structure that is capable of work as a transducer, meaning it will receive different types of energy such as sound and vibration, for example, and will transform that into a nerve stimulus that eventually is going to be transferred to the brain. The receptors can have multiple different types of classifications, such as based on the modality. They can be receptors for temperature, receptors for pain, receptors for light, receptors for chemicals, based on the origin of the stimulus, which can be stereoreceptors, interoreceptors, or proprioceptors. The muscle spindles are proprioceptors at the level of the muscle senses. The receptor for the general senses are those located all over the skin. There are two main kinds or two main groups. These receptors are going to be unencapsulated, as you can see right here, free nerve endings, and encapsulated, as you can see right there, tactile corpuscle for light touch. Somatosensory pathways. Pain is an example. Right here in the finger, you're going to stimulate the receptor. The receptor sends the information up to the brain using the sensory nerve. Now, the only part that you need to pay attention now is that it's not just one nerve, but there's actually three. Here you have the first nerve gets in contact with the second nerve in the spinal cord right here. It makes a synapse. The second nerve goes up, makes a synapse in the thalamus, and the third order neuron takes information up to your brain. Referred pain. This happens because when people have heart attacks, they feel pain in the left arm because the first order neuron for the skin goes this way and makes a synapse with a second order neuron. But what happens is that the heart, the first order neuron also makes synapse with the same second order neuron. So when the brain receives the information, it's not sure whether it's receiving the information from the skin in the left arm or in the heart. So that's why you feel pain from different areas. Chemical senses. Taste. The sense for taste is located in the tongue. The tongue is not smooth like the screen in your computer, but rather has a whole bunch of rough surfaces, because in your tongue what you have are going to be papillae. Inside the papillae is where you have the taste buds. We have valate papillae, fungiform papillae, foliate papillae, and filiform papillae. Taste buds that we have, most of them are located in the valate papillae. What type of flavors we can identify? Salty, sweet, sour, bitter, and umami. Those are the main flavors. Cranial nerves that work with the tongue. We have cranial nerve number 7, number 9, number 10. So those are the ones necessary to send information up to your brain from your tongue so we can receive information regarding taste. Smell. The receptors are located inside your nose, and the nerve that is going to take the information up to your brain is going to be cranial nerve number 1. So basically what happens in here, other particles that are in the air, they go inside your nose. So these other particles are going to get trapped in the mucus that we have in our nose. When they get trapped in the mucus, these other particles dissolve and they release the chemicals that they have. And these cells are going to be the receptors that are going to capture these chemicals, and they transfer the information up into your olfactory bulb. From the olfactory bulb to the primary olfactory cortex right here in brown. But you see they are associated with other areas because smells are usually associated with sensations. If you smell gravy and turkey, that brings memories about family and your best friends. Hearing equilibrium. Sound is the vibration of molecules that we can hear. Now, sound has certain qualities such as loudness. Anatomy of the ear, we're going to see that we have an outer ear, middle ear, and inner ear. The outer ears, we have the lobule right here at the bottom, we have the helix, we have the auditory canal, and it ends in the tympanic membrane. The middle ear goes from the tympanic membrane to where this structure right here, which is going to be the vestibule, is going to start. So if we talk about the middle ear starts basically in the tympanic membrane, continues with these three little bones. They are called malleus, incus, and stapes. All three little bones are called ossicles. And in the middle ear, we have this structure, which is going to be the auditory tube that goes down into basically the back of your mouth. Helps release air pressure that we may get in the middle ear. Then we have the inner ear. In the inner ear, we have three main structures. The cochlea, this is a vestibule, and this one right here are the semicircular canals. The cochlea is going to help you with hearing. Vestibule and semicircular canals are going to help us with equilibrium. These yellow areas are showing you the nerve, which is going to be cranial nerve number 8, the vestibulocochlear nerve that is going to help us with hearing and equilibrium. You can see a cut of the cochlea where you have three tubes. This one on the top is going to be called scala vestibulae. The one on the bottom is going to be called scala tympani, and both of them are filled with perilymph. The one in the middle is called the scala media, or the cochlear duct, and this one is filled with endolymph. In the scala media, we have the organ of Corti, where you find the tectorial membrane, and you have the hair cells. The hair cells are going to be responsible for sending the information to your brain to hear. So we are able to hear because we have the organ of Corti. The trick is to move the hair cells right here. In order to do that, we need to move the basilar membrane. When we move the basilar membrane, the hair cells are going to move because the tectorial membrane is basically fixed. For example, we produce sound waves with our vocal cords. The sound waves go through the air and they go inside our auditory canal. They bounce against the tympanic membrane. The tympanic membrane vibrates. That vibration is transmitted to the malleus, intes, and stapes, and the stapes is connected to the oval window that is going to transfer that vibration inside your cochlea right here. Now, in the cochlea, the vibration of the three little bones is transferred inside into the fluid in the form of waves. The waves are going to go through the perilymph right here, and that perilymph is going to move the vestibular membrane. The vestibular membrane moves the endolymph in the cochlear duct. The endolymph moves the basilar membrane. By moving the basilar membrane, we are going to move the hairs. The hairs open channels for potassium, and that initiates a depolarization that sends the information to your brain using this nerve, which is going to be the vestibular cochlear nerve. The basilar membrane is stimulated in different areas to allow us to hear
different sounds.
The vestibular cochlear nerve to send information to the brainstem, from the brainstem to the thalamus, and from the thalamus into the primary cortex auditory area. Our next topic is equilibrium. There are two main types of equilibrium, static equilibrium and dynamic equilibrium. The macula is for static equilibrium. Semicircular canals are for dynamic equilibrium. In the vestibule, we're going to have structures called macula. If you look at this figure right here, what we need to do is we need to move the hair cells. For example, when you're inside your car, you don't move, but the car moves. The car movement will move your macula, utricle or saccule, and then this gel right here will lag behind, and because it lags behind, it will move the hairs. And when it moves the hairs, transfer information to your brain. The semicircular canals. In this case, semicircular canals is when you are turning. For example, when you are sitting down on a chair and that chair starts turning around, then that's when you use the semicircular canals. The semicircular canals have a gel. In this case, it's called crista ampullaris. And inside the gel, you have hair cells. What happens is that when you turn, the fluid inside this tube is going to push the crista ampullaris, and then this pushing moves the hair cells, and when the hairs move, they send the information to your brainstem, from there to the thalamus, and from there into the vestibular cortex that is going to help us maintain equilibrium. Don't forget that we have three different areas to maintain equilibrium. One of them is the eyes, the other one is the cerebellum, and the last one is going to be the ears. Vision, we are able to see light. And vision is the perception of objects in the environment by means of the light that they are going to emit or reflect. If there is no light, we cannot see. So right here you have the eyes. The white part is the sclera. The color of your eyes is going to be the iris, and pupil, the black part in the middle. The iris and the pupil are covered by this crystal and translucent area called the cornea. Also, right, we have the lacrimal glands. The lacrimal glands produce a lot of tears to clean your eyes. It also has substances such as immunoglobulin A in order to defend us. The tears come out of the eyes through the lacrimal sac into the nose through the nasolacrimal duct. In addition to those structures, we are going to have muscles associated with the eyes. So this is superior rectus down here, inferior rectus, medial rectus, lateral rectus. On the top is superior oblique, and another oblique at the bottom, inferior oblique. This muscle moves due to the fact that they are associated with nerves. In the case of superior oblique, it's the trochlear nerve. Lateral rectus is the abducing nerves, and all the others are going to be moved with the oculomotor nerve. We can see the anatomy of the eyes after the eyes have been cut, so you can see the inside. So there is a space called the anterior chamber between your cornea and the lens, where you're going to have the aqueous humor. Behind the lens, you have this other space where you're going to have the vitreous humor. This is the wall of your eye. Okay, it has 3 layers: external, middle, and inner layer. They are called tunics as well. This external area is going to be the sclera. The middle area, so-called the uvea, is going to have a lot of blood vessels, and it has 3 main parts: the choroid, which is this part in brown. It continues with the ciliary body, which is going to be the second part, and the third part is going to be the iris. The ciliary body produces this muscular mass that is going to be able to hold the lens in place using the suspensory ligaments. So what's the job of the lens? The lens sits behind the pupil, and it allows to adjust the light before it reaches the back of your eyes. So the light goes in through your cornea, through your lens, vitreous humor, and goes to the back of your eyes. The inner layer, right here in yellow, is what we're going to call the retina. The retina is where we have the receptors that will allow us to see. In your retina, this is the area where the nerve is, and this area right here is the area that will allow us to see detail. So where the nerve is located is called the optic disc. This area is orange, and this will allow us to see everything. This is what we consider the peripheral vision. This area where all these receptors are going to concentrate, they're going to be the macula lutea. The very dark area is going to be the fovea centralis, that is going to give us the detailed vision, right? The ability, for example, to read. So this is peripheral vision. This is visual acuity. So these are the receptors that you have in your retina: the rods and the cones. Cones is for a lot of light. Rods are for a little bit of light. Cones are, for example, during a sunny day, and rods are, for example, for spaces where there's not a lot of light. If there is no light, we cannot see. In addition to that, there are other cells such as ganglion cells and bipolar cells. Some people can read, for example, but they cannot identify colors. There is a test called Ishihara's test for that purpose.